Yep, it's already in SMCE. So I'm
Great.
gonna actually have you guys kind of walk through it with me as I'm going. We'll go fairly quickly unless you have any questions because I know you went over a lot of this stuff last semester. So I guess first, can you guys see my screen?
Yeah.
Yes.
Okay, awesome. Let's start with you logging into, so I'm already logged into SMCE. So why don't we start with having you log into SMCE? And I do want to point out that everything that I walk you guys through today, you don't have to like write down or anything because I have all of this available in the GitHub repository. So I created a new subdirectory in the GitHub repository for the training content within the RNA-seq DGE Jupyter Notebook subdirectory. And this directory is called RNA-seq DGE JN for Jupyter Notebook _ anyGLDS. So saying any GLDS data set that you could put in there. So I put the link in the chat. And pretty much these are the instructions that I'm going to follow. And you guys are going to follow along with me so that you know how to set this up on the SMCE system. So very similar to how you started the content last semester is this is just describing the two files that we're going to be running on SMCE. And so hopping down to the getting started section, the first one would be to go to the Dask Hub website, and I'll put this in the chat too, and log on to SMCE using your username and password. And then once you're logged on there, whereas before you selected the small server on demand for this one, because I actually added in an additional visualization because I know that hopefully you'll be able to put this stuff together to submit an abstract for a conference. So I wanted to give you another way to view and interpret the data, and it requires slightly more memory than the original Jupyter Notebook. So you'll have to select the large server on demand this time. So let me know when you have that sprung up. And it'll look similar to this. Of course, this would be your only tab on there.
So Amanda, I just want to make it clear, you want us to log on to SMCE right now, right?
Yeah. Yeah,
Okay.
I just want to make sure everything works for you guys. So we'll do an example and we'll do the GLDS 104 data set that you guys went through last time using this new notebook. And I just want to make sure everything works for you, while I'm here. So
Okay.
if you guys run into any issues on your end, I'll be able to help troubleshoot, hopefully.
Okay, I will try.
Yeah, so the only difference is just this time, make sure you're selecting the large server on demand.
So I need to log off. I think before the meeting, I log on to the small. So let me log out.
Okay. You can actually you don't have to log out. You can go to File, Hub Control Panel, and you can stop your server. And then after you stop your server, it'll show up a button that looks like this. Oh, it's in the troubleshooting. That'll ask you to start your server again, and then you could start your server, and it'll bring you to that same selection menu.
Okay.
And then except when you start your new server, make sure you're starting the large
Okay.
server on demand. And then let me know when you guys are waiting for that to spin up, because I do want to just talk about, if you'll notice, our repository has changed a little bit in the recent months. And so I want to quickly go over that stuff with you guys.
Yeah, I'm
So we
just
can do.
waiting for it to log in now.
Okay, great.
Yeah, mine is running halfway too.
Awesome. Okay, so while you're waiting for your servers to spin up, so you guys might have noticed that some things have changed in the GeneLab repository. So we actually have merged with one of our sister repositories called the Ames Life Sciences Data Archive. And I don't know how much of my intro lecture you listened to, but I talk a little bit in there about the Ames Life Sciences Data Archive. So that archive actually hosted all non-omics data from space relevant studies. So let's say that, an experiment was run and one of the analyses that was done is they looked at bone density, right? So bone density is a physiological metric. It's not an omics data. And so that physiological data would be submitted to the Ames Life Sciences Data Repository as opposed to GeneLab. So before, these were two separate repositories, and the ALSDA one was not as streamlined or normalized. So there would be like different terminologies to mean the same thing. And so we're really trying to make the Ames Life Science Data Archive follow the same structure as GeneLab, where we use the same terminology when we mean the same things to help make it more machine readable and easier to navigate and understand what information is there. And so when we brought these two repositories together, we called it the Open Science Data Repository. So this is all of the open science, life sciences that we have at NASA. And that the website that I put in the chat, osdr.nasa.gov/bio/repo, that will bring you here. And this should look pretty familiar. So it's set up very similar to the original GeneLab repository, except this time now you'll notice in the search filters, whereas before we had GeneLab and some of these other omics databases that we would federate data from, or you could search those databases while you search GeneLab as well. Now we have another one here, ALSDA. So now you're able to search in ALSDA as well when you type in search terms here. So for you, of course, you're going to want to use GeneLab data, so you don't have to click the ALSDA filter. You could just have GeneLab data sets. And so this will only be looking for data sets that have GeneLab data or omics data. And then depending on what you're interested in, so if you're interested specifically in a mission, so a mission being a spaceflight study that was run on a NASA mission, then you would click mission, or if you just want to search any study that's in here, you could leave it just to search a study. And then we have additional study filters here. So here, if you wanted to only look for spaceflight missions, you would filter spaceflight. If you didn't care and you wanted to look at both spaceflight and then the ground-based experiments where we mimic some aspect of the space environment, so whether it be like radiation exposure or mimicked microgravity, those are considered ground studies. So you can modify your search filters depending on what you're interested in. And then for the assay type, you're going to want to have RNA sequencing data, right? So we're going to be analyzing RNA-seq data with this notebook, so it makes sense to only look for data sets that have RNA sequencing data.
Yeah.
And then if you are interested in a specific organism, this is where you could look. So if you only want to do and generate data from rodents, then you would come in here and select rodents. If you only want to look at human data, plant data, etc. So based on what you're deciding that you want to do your research on, you can pick and choose filter based on the organism. And so once you do that, I'll filter based on rodent, you'll have a variety of studies. So you'll notice that these studies now, they have an OSD identifier that stands for Open Science Data. And that's because any given study can have GeneLab data, so that's omics data, or it can have physiological data, that would be ALSDA data, or it could have both. So data sets that have GeneLab data, when you click on it, and we're going to go to, so this is the one thing I should back up for a minute. So this Jupyter Notebook starts with data sets that have raw counts data. We have not processed all of our RNA-seq data sets yet, and so there are data sets that don't have raw counts data. Unfortunately, we have not implemented a filter yet where you could just filter and only have data sets that have processed data. So that is one limitation. It is on our to-do list, so it will get done eventually, but not likely in time for you to be able to use it for this. So this is going to be a little bit tricky because you're going to have to go in, like when you pick some data sets, you'll have to go in and see if there's actually the raw counts files there or not. And you'll only be able to use data sets that have those raw counts files.
Yeah, I already kind of took a look at that, and I did find a couple data sets that have the stuff needed, so it's okay.
Excellent. Okay, great. And so I'm just going to go for OSD 104, which is the data set that we worked on in the training that you took last semester. And so then, just like anything else, you click on the data set and it'd bring you to the landing page for that data set, which I know you're familiar with. So as I mentioned before, the initial identifier now is this OSD number. And then if it has GeneLab data, like this one does, it'll also have the GeneLab ID, and that'll be the GLDS number. So you'll see that right at the top, right under the image here. Why this is important is because especially with our newer data sets, these numbers are not the same anymore. So for a lot of the data sets that we already had published, because we only had GeneLab data in there, the OSD number matches the GLDS number. However, we've recently uploaded data for new studies that only have physiological data and not GeneLab data. And so that OSD number would go up where the GeneLab number would remain the same. So for newer data sets, these numbers are out of sync. And so I'm pointing this out because in the notebook, we're actually going to want to make sure that we indicate both the OSD number and the GLDS number so that we ensure that we're getting the information from the right data set. And so for this one, of course, it's the same. So OSD is 104 and GLDS is also 104. And then since I know you've gone through this, just to know, you go under files and then GeneLab, if it has GeneLab processed RNA-seq files, pretty confident that you're going to have the files that you need in here. So you probably don't have to go further than this. So if you don't see GeneLab processed RNA-seq files in the files list, that means it hasn't been processed yet and you won't be able to use the notebook for it. But if you really want to double check and make sure, you can even click on the GeneLab processed data files, and you're going to want to go to raw counts data. And what you're looking for is to make sure that it has these files that end in genes.results. So if you recall from last semester, this is the raw counts data that's generated by the RSEM tool that we use to quantitate our data after we map it back to the reference genome. And so these are the files that this notebook will start with. Any questions on navigating the new system?
I just want to be reminded, the raw counts data are already being filtered, right? Normalized.
No, we are going to do the normalization. So this is some of
Oh,
the things that are
last
going to
time I
be.
thought you want us to get the counts table. So not, oh, that's exciting.
Yeah, so this is going to be a lot of fun. It's going to be a little bit different than because last time we kind of imported a table already.
Mm-hmm.
Whereas this, we're actually going to start with these files and pull them in.
Ah, because I was keeping asking Romar to find a table, maybe a CSV type table. So
Yeah, so I
Romar, are
changed
we
it a
going to
little
do
bit,
more this time?
Yeah,
little bit more.
I kind of figured out while I was looking at the data sets this week. I had to search around again to look for the correct files. I did find a data set now that has
Okay,
got everything we need. So
great. Yeah, so what you look for are these genes out. As long as it has these genes.results files in it, you'll be able to run this modified Jupyter notebook.
Right.
So, oh, go ahead.
Oh, no.
Are you going?
Thank you.
So do you guys have this up now?
yeah,
And Dr. Lin, you too?
Yes.
Okay, great. So similar to what you guys did in your course last semester, you're going to first start by clicking on the terminal. And I do want to point out again, this is all detailed on GitHub. So after you do your on-demand server, it tells you to select the terminal in the menu. So you don't have to write this stuff down. It's all here in GitHub. And then once you select the terminal, similar to what you did last time, except this time we're going to use a slightly different command because we're copying different files. So we're going to copy and paste this command in the terminal, and I'm going to put this in the chat for you guys.
And
So
so
when you,
for some reason my chat is muted, and I did go to the setting to unmute it. It is still
yeah.
not working.
I can't see the chat right now either.
are you, okay, so in that case, if you guys want to go to the GeneLab training GitHub repository, so if you just, go to Google and Google NASA GeneLab training GitHub, you should have the link to this. It's
Okay, let's see.
Git, yeah. So let me know when you're seeing this. It's github.com/NASA/GeneLab-training.
Yeah, I got it.
Is that the same one we did last year, right? I mean,
Yeah. Yep. So I added to it. Yeah.
Yep, I'm in.
Dr. Lina, are you seeing this too?
okay. Let me just make sure.
Yeah, so if you're there, you should be seeing, this is the main landing page for the GeneLab training. So if you just go github.com/NASA/GeneLab
GeneLab
training.
training. Yes, I'm on that page.
Perfect.
And
So now,
under
yeah, so now
GeneLab
you can,
4.
yeah, so now you go to GeneLab4U, and then you'll click on RNA-seq, and then you click on the June 2022 Educator Pilot directory. And then from there, you're going to click on the RNA-seq DGE JN, so the differential gene expression Jupyter notebook. And then in here, you'll see a new subdirectory that wasn't here before. This is the one that I added. It's called RNA-seq_DGE_JN_anyGLDS. So you want to click in that folder, and then these are the instructions that I'm following. And
Okay.
so we are down here in the getting started section, and you guys spun up your large server on demand, and you opened up a terminal in your SMCE instance. And so now the next thing would be in step 4, there is a command there for you to copy and paste. So you just want to copy that on the GitHub repository, and then go back to your terminal on SMCE, and then paste it and press enter. And after you press enter, what you should see are 2 files showing up in your home directory that weren't there before. So on the left side, you should see download_file_urls.py, and you should see RNA-seq_DGE_JN_anyGLDS, and then I just have last month, which is when I finished this, and then the year.
Okay. I got
You
it.
guys are both seeing that in your home directories?
Yes.
Awesome. So the next thing you do, again, following these instructions. So it just shows you should see these 2 files, and then you want to double click on the one that says RNA-seq DGE JN. So you'll double click on this one, and that will bring up a notebook that looks like this. Both seeing that?
Yes.
Yep.
Okay, great. And so now this is very similar to the notebooks that you ran last time. So similar thing, you have a mix of these markdown blocks, which give you instructions, and then these code blocks, which you'll be able to run. And so a slight difference here is we have updated our pipeline since doing the educator bootcamp. And so we're using pipeline version F, which basically has some updated tools. And we actually pull in annotations using a slightly different way, because we create an annotations file kind of separately now and just pull in that annotations file. So those are pretty much the main differences here. You'll notice the same DGE workflow. So we're still going to be pulling in raw counts, except this time it's going to be in the form of those genes.results files that you see on the repository. We're still going to be pulling in sample metadata, except this time, rather than it being a file that I created for a specific data set, we're going to be able to upload sample metadata from any GLDS data set that has the raw counts, using this download file, py file that also went into there, which is just a script that'll help us do that. And so we're still going to be doing the same thing. So you're still going to generate a raw counts PCA plot. Then you're going to go ahead and you're going to normalize those raw counts using DESeq2. You're going to generate a normalized counts PCA plot. You're going to do your estimate your gene dispersions to help further correct your data, and then you're going to run your hypothesis testing to identify differentially expressed genes. And then we're going to put all that information together in a differential gene expression table, and then we're going to add on gene annotation. So instead of it having ensemble IDs that no one knows off the top of their head, we're going to have actual gene names and gene symbols like you did last time. And then we're going to run visualization, which is, the most fun part of this notebook. And so again, generating a DGE PCA plot, the volcano plot that you saw from last semester, the heat map that you saw from last semester, except this time I also added on a gene set enrichment analysis. So we'll talk a little bit about that at the end.
So,
yep.
And so the annotation, so the file, the gene results doesn't have, it's only have raw counts, right? It doesn't have the annotated file. So annotation happened at 4B, does that mean we need to upload the library?
We actually will pull in a table that we create now. So instead of actually, so whereas before, if you remember, we went through a series of steps where we added in annotations from bioconductor for that specific organism, and then we use those IDs to add in string IDs, so information about the protein analysis, and then we added in information about gene ontologies, and we kind of went through all these iterations where we were building it up. Now, actually, in our official GeneLab data processing repository, we actually packaged, because we use annotations in a variety of our omics data, not just in RNA-seq. And so we decided, instead of running this every time we run a different omics, let's just pull out the annotations, make a separate table that we can use to link back into any omics data type. And so we have this GeneLab reference annotations pipeline that we now use. And so this one, and there's links to this in the DGE Jupyter notebook. So when we get to that part, you'll see all the links for this. But basically what this has is it has what's called an annotations CSV file that we generated. And then for any organism that we currently have in our repository, we have the name here, species, if there's a substrain or a strain, like for our microbes data, the version of ensemble, which is the main IDs that we use for this, the reference source, because there's a few different types of ensemble databases, where we get the FASTA file from. So that's what you mapped back to in the FASTQ to Counts Jupyter notebook last semester. And then the GTF annotation file that tells you where different regions are within the genome. And then the taxon, so this is specifically needed for finding the bioconductor annotation package, which we used to add in steps and thing. And then we upload that final table to a Figshare link. And so this Figshare link, take a look at the one for mouse. And there's links to all this in the notebook, so I'll point that out when we get to there. So
So does that mean the file is being uploaded already, or we still need to use the table to upload the file?
we are going to be pulling in the table, right? Because we don't know, it depends on what organism you want to use. So I don't know yet which file you're going to want to use. This is the mouse one looks like. So this should look familiar from last semester, right? Where we pulled in, we have the ensemble ID, the symbol, gene name, refseq. So now we already have this file already made. So basically in the notebook, when we get to the annotation step, what we're going to do is, depending on which organism you select, it'll pick that appropriate table and pull in these columns from there.
Okay,
So it makes
that's
it a
convenient.
little bit simpler. Yeah.
Great, thank you.
Yeah, for sure. And so in here again, have a table of contents. So the biggest difference between what you did last semester and this new one is how we set up the metadata in the results files are going to be different, right? Because we need to modify this so that way any data set that has those genes.results files you'll be able to use. And so you'll see the table of contents have slightly different steps in it at the beginning, but then all of these steps towards the end from 3 on will look very familiar from last semester. And so, and all of these link to the respective sections. So if you ever need to come back, you do that. The other awesome thing that I did this time around is, before, if you remember, we were going back and forth between a Python kernel and an R kernel. So we were running things in using Python language or an R language, but when we were running things in Python, we were actually running bash or Unix commands, but we were doing it from the Python kernels. We had to add that exclamation point in front of things and sometimes weird symbols. Well, turns out that you can actually incorporate a bash kernel into Jupyter. And so that completely got rid of having to do anything weird because we're trying to run bash commands with Python. So now we actually have a bash specific kernel. So you won't have to deal with any more adding exclamation points in front of your commands, which is really nice. And this is also pointed out in the instructions on GitHub. So you'll see here that after you open up your file, it says you're now ready to run it. And then it tells you which steps you need which kernel for. So the first 2 major steps, so 0 and 1, you're going to be running the bash conda environment. And so this is instructions for where to find that and then how to change it if you don't have it. So we'll go over that now. So you want to look in your top right corner of your Jupyter notebook, and it should say bash conda environment GL4U-R-2023. If it doesn't say that, then what you would do is click on here, and that would bring up this drop-down menu. And then in the drop-down menu, you would search for where it says bash conda environment GL4U 2023. And again, this is all in the GitHub repository with pictures, so you don't have to like memorize anything.
Mm-hmm.
And so you go ahead and click that, and then you would click select, and then you just want to make sure that up here you see that it's running that specific environment. So this environment is telling the system, hey, we're going to be running Unix commands in a language, in an interpreter called bash. So Unix, there's a variety of different interpreters, which I think you guys went over in last semester. We use the bash interpreter. And so now we can go ahead and start running this. And I do also have a note here right at the top that says that it uses both bash and R kernels. So I have at the beginning of each step which kernel you should be using. And so you just want to pay attention to that, right? And it also, again, is written in the GitHub. So step 0 and 1 use bash. Steps 2 through 5 use the updated R kernel. So we'll get to that when we get to those steps. So the first thing you want to do here is this is where you input your specific OSD identifier and the GLDS identifier. So for this, I just am using OSD and GLDS 104. And so basically, if for whatever data set that you selected, Ramar, what you would do is just replace this number with the number of the data set, right? So if you want to use like 101, then you replace it with 101. Make sure you're using the right corresponding GLDS number. And again, just as a reminder, that information will be right at the top. So the OSD identifier first, just under there would be the GLDS identifier. So you want to make sure that you're updating these based on which data set you're running. So again, we're going to do 104. So what this is doing, as you remember from last semester in bash, is it's creating a variable. So this is creating a variable for OSD, and it's creating a variable for GLDS for the respective data set you're using. And so you'd go ahead and run that. And then similar to what you're used to, you would see a star right next to it when it's running. When it's done running, you'll see a number. And so now these next steps should look familiar as well, except this time we're using variables. So we're using the variables that we just set up here, and we're going to start by making our top level directory. So if you remember, I really like to keep data organized when we're doing RNA-seq analysis because we generate so many files. So I set up a directory structure at the start of processing any data set. So if you go ahead and run the first one, you should see a top level directory created here with your OSD number and underscore followed by your GLDS number. So you would see it on your left here. Once you do that, now within that directory, we're going to make a series of subdirectories, which is where we're going to store the process data that we generate. So the first one, we're going to generate a subdirectory for our metadata. So you'd go ahead and run that. And then we'll generate a subdirectory for our RSEM counts data, which we're going to pull in from the repository. So we'll go ahead and run that, and we'll take a look at all these after we run them. And then we're going to create a subdirectory for our normalized counts, right? Because we're pulling in the RSEM raw counts. So one of the things we're going to be doing is normalizing those. So we'll create a directory to hold the normalized counts. And then within there, because we create PCA plots of the unnormalized normalized counts, we have a subdirectory of where the PCA plots will be put. And then we're going to create a series of subdirectories for the differential expression data. So the first one is going to be our top level one, where everything related to differential expression is going to go. And then we have a series of subdirectories. One is going to store all of the plots that we're going to generate. And then we have a subdirectory for each specific type of plot that we're going to make. So we'll go ahead and run this. And then if you want to see what that looks like or make sure that you did it, you can go to the left, double click on that top level folder that you made. You should see now the 4 initial subdirectories that you made. And then you can kind of go into the RSEM one. We don't have anything other than that, but in like 04, we have the PCA directory that we made. And then to go back, you can click on it up here in your path. So now if I wanted to go back one, we could do that. And then if you look at the DGE folder, we have this DGE plot subdirectory in here, and that'll be all the subdirectories we made for each respective plot. So now before you continue, make sure you go back to your home directory. For me, I actually have a separate home directory, but you should just be this one folder and then a / is where you should be. You won't see this home. I did that just to keep things clean because I have a lot of stuff in my home directory. So once you're here, make sure you're seeing this on the left. We can continue on in the notebook. So we created all of our directories and we're ready to start setting up variables for those directories. So again, this should be familiar from last semester. Now that we made all these directories, let's set them all up as variables. So anytime we need to put anything in any one of those directories, we don't have to write out the full path. We can instead just use a variable to represent that path. So that's what this step is doing here. So you just run that, and then it's storing, remember, in memory, what each one of these variables mean. And they're just pointing to a specific location in that directory structure. So now what we're going to do here, and everything is explained in here in text. I'm not going to go through this in horrible detail. Of course, go ahead and read through all of this. And if you have any questions, let me know. So what we're going to do here is we're using this script called download files URLs PY. That's one of the things that you added in here. If later on you want to take a look at that script is written in Python, you're more than welcome to. It's a little out of scope, but if you're curious, it's there, you could look at it. So we're going to run that script. And what that script takes in is 2 pieces of information. It needs to take in the OSD number of the data set that you're interested in, which we stored in a variable called OSD at the start. And then it wants to know what type of file do you want to pull in. And so all of the metadata that you see for a data set in here. So everything about the samples, if you scroll down in the repository, you'll see a samples table here, right? And this gives you information about each one of the samples in the data set and a variety of different columns telling you what different aspects, right? If they were part of the ground control group or part of the space flight group, if they had a specific light cycle, all sorts of information is going to be associated with these samples for each study. The other tables that we have in here is called the assay table. Some data sets have multiple assays, and that just means that the samples from that study were used to look at different things. So for instance, for this data set, we did DNA methylation profiling as well as transcription. And so we would the transcription profiling table is what we're going to be interested in. In this here, again, is just a table with information about what was done with these samples to prepare them for RNA sequencing. So this is things, if you remember going through that really big RNA-seq overview, we did things like taking a look at the libraries, right? So we took a look at the libraries, looked at the quality of the libraries, including the RNA integrity number. So all the information from that library preparation for preparing these samples for sequencing is put in a column in this assay table. And so like what type of library preparation, if you remember, there's 2 ways you could prepare libraries. You could either remove all ribosomal RNA, or you can positively select for RNA that has a poly A tail, which would be your message RNA. So this is a ribodeplete study. So it's prepared using ribodeplete. If you remember, you can sequence your samples in one direction, or you can sequence them in one direction and then back the other way. So either single end or paired end. So information like that will be stored in this table. You can use a library prep kit that'll generate stranded data, so keep track of which strand the RNA was generated from, or there's also unstranded data where it doesn't keep track of which strand the RNA came from. And so like all that information about how these samples were prepared is going to be stored in this assay table. In addition to that, there's also columns that'll say where to find the respective process data files. So this is going to be important because remember, we want to make sure that we are going to pull in the genes.results files, right? And so that information is all stored in these tables. These tables on the back end of it is in a format called ISA. It stands for Investigation Study Assay or Investigation Sample Assay. And so if you were to go down into your files section here, and if you were to look at the study metadata files, what you see is this file that has the GLDS number. It tells you its metadata. Sometimes it also has a GLDS number duplicated, and it ends in isa.zip. So any information that's stored in that sample table or in that assay table is going to be one of the files inside this is.zip file. And so what this script is doing is we're telling it, okay, I want you to run this script, which is going to, and that script is set up to be able to pull information from the repository. And so it wants to know, the script first needs to know what study do you want to use? And that's for the OSD identifier. And then it says, what files do you want to pull in? And so that's why we're specifying that is a .zip file, because that contains all of the metadata or data associated with each sample. And then then the last argument here is it's asking you, okay, now once I download the file that you requested, where do you want me to put that file? And so remember, we created a metadata subdirectory to hold all the metadata. And so in this command, we're running this script to download files from the repository. We're letting it know which data set we want to pull from. We're letting it know which type of files we want to download. And then we're telling it where to put the files that are downloaded. So that's what this code block is set up to do. So then you can go ahead and run that. And you will see it'll tell you information, and it'll tell you where it's pulling the file from, right? So you'll see that this is basically the link to where that is.zip file lives on the internet. And then this is running a curl command, which is one of several different commands you can use to pull files from the internet. And this is just basically more information about how quickly it's downloading the file and stuff like that. And so once that's done running, it'll run pretty quickly. We want to look inside that metadata directory because remember, that's where we told it to download this file. And so we use the ls command, which again should be familiar from last semester. So we're going to list everything that's in that metadata directory. And what we should see is that is a .zip file named exactly how we saw it in the repository. And so that's good. Confirm that we have our metadata. And so this next thing that we're doing here is we're creating something that we're going to call a run sheet. And so if you remember, when I went over this assay table and the sample table, there's a ton of information in here. We don't need all of it to run differential expression analysis, right? The things we need to run differential expression analysis, we need to know which samples belong to which groups. And the groups are determined based on which factors they have, right? Or which variables were tested. So things like spaceflight would be a factor, right? If this was a radiation exposure study, things like radiation or not irradiation would be a factor. So, and the other thing that we need to know is for each sample, we're going to need to know where that genes.results file is, right? We're going to need to know where the files are that we're going to have to pull in. So that's what this function does. It's in a tool called DP Tools, which was actually created by Jonathan on our team. So he created a program called DP Tools to be able to take the information in this is a file, which is everything you see on the repository, and kind of pick and choose what's going to be relevant to do differential gene expression. So what this argument takes in is, again, it wants to know what data set you're doing this on. It wants to know what type of data you're doing this on. So this is bulk RNA-seq because we actually parse the data for our other omics data types as well. And so depending on which omics data type, you might need different information. And so we want to tell it that it's bulk RNA-seq. And then because we have different versions of data sets in our repository, so if you'll notice at the top, this is on version 4. And this is because we'll like update things. So if we notice that there's a typo, or if we notice that we didn't have some metadata that we maybe should have had, we'll update our data sets accordingly. And so anytime we update it, we release a new version of that data set. So what this is saying is what version of the data set are you working on? And we just have an option here called latest, which means give me the most recent one. And then the next thing it wants to know is where is the is.zip file that this tool is going to be parsing. And so from the previous commands, we downloaded that is.zip file in the metadata directory, and it's always going to end in is.zip. So we have this wildcard here. So that means no matter what data set you have, as long as it ends in is.zip, which they all do, we'll be specifying that file. And now this function doesn't let you direct the output run sheet somewhere. And so we just have this separate command here, which is a move command, which should also be similar from last semester. And so we're going to move the run sheet that's generated into our metadata directory to keep all the metadata information in the same place. So we can go ahead and run that. And this, again, you're going to see some information about where it's pulling information from. And it'll run through. It shouldn't take too long. So mine's already done. Again, make sure there's a number at the side of your code block, which indicates that it ran. And then if you ever get any errors for any of these, just shoot me a message, shoot me an email, let me know. Hopefully you won't. And then it tells you a little bit of information about what it just created, right? So it created the run sheet, and this is the name of the run sheet that it created. And it tells you how many rows and how many columns the run sheet has. And so the next thing that we want to do here is take a look at our metadata directory and make sure we see that run sheet in there. So we can go ahead and press play. And if you remember, you should see your is.zip file that you downloaded previously. And now you should also see a file that ends in runsheet.csv. And so this is another thing that's going to be important. We're going to want to make sure that we know what the exact name of this runsheet.csv file is, because that's something we're going to have to check in when we bring that run sheet into R when we do DEC2. And so just in case you guys were interested in what information was parsed and pulled and put into this run sheet, this next block here uses a command that I know you're probably not familiar with. So there's several nice commands within the Unix environment that you could use to help looking at files be nicer. And so if you remember from last semester, you would look at files using a cat command, which is great, but sometimes, like for this, we have a bunch of different columns. So maybe kind of separating out the different columns would make it easier to view. So this is just running a column command, and it's separating things out based on tabs. So it's tabbing things over for each column. And then again, you're doing this on your run sheet file that's in your metadata directory. So just take a look at that. And I know it's not like the prettiest, but so these are the column titles. We'll have the sample name. And so you'll see this is the first sample name in here. Another piece of information we need to know, which I know that we didn't go over last semester, is if it has an ERCC spike in. And so in RNA-seq data, there's a variety of different quality control things that you can do. One of them is adding a spike in of RNA that was synthetically made, so made in a laboratory where you know the sequence and you know the concentration of it. And the idea is it can help you with a variety of different QC metrics. And so at GeneLab, depending on whether or not the data set has ERCC spike in, kind of changes how we process them. But for you guys, we're not going to worry about ERCC or not. I just wanted to point out what that column is. The next column, so this is a true false column. So that's why you see the false here next to each sample, because this did not have ERCC added to them. The next column here is the organism. So this will tell you which organism your samples were generated from. So of course, this is a mouse study, as we know from last semester. So this would be a mouse study. We need to know if the data was paired end or single end. And so that's another piece of information it pulls in. So it asks, is it paired end? And then it's a true false here. And then it tells you information about where to find the files. So this is where to find the raw read1 file. This is where to find the raw read2 file. And that's because usually when we use this run sheet, we use it to process data sets from start to finish. So starting with the raw files, we just don't have the compute resources to do it here. So that's why we're doing things a little bit differently. That's the information that you see in this run sheet. The next column is called factor value. So again, depending on how the study is set up, this study is fairly simple. It has one factor: either animals were flown in space or they were kept on the ground. Nothing else was done to them. So there's only one factor value here for spaceflight. And you'll see for this column, it either says ground control. I know it's kind of cut off here. So this sample was a ground control, or for some of the later samples, you'll see it says spaceflight. So basically for this factor column, it says that. Now, some data sets you'll notice have a lot of factors. So maybe it was a ground-based study and they looked at both radiation exposure and mimicked microgravity. And so you'll have one factor of whether or not it was exposed to radiation and another factor of whether or not it had simulated microgravity. And so if that's the case, you would have two columns here, one for each factor. So depending on how many factors or how many things were investigated in your study will determine how many factor columns you have. And that's going to be important because when we group these things to run differential gene expression, we want to group all the samples together that were exposed to all the same factors, right? So in this case, we just have spaceflight or ground control. So we're going to use information in this column to know if the sample belongs in the spaceflight group or if it belongs in the ground control group. And then another piece of information here, this again is a GeneLab specific thing because not only do we generate our own data sets, we also take in data sets from other repositories. And so when we take them in, they might, the sample might have had a different name than how the name ends up on GeneLab. So we like to keep track of that. So that's why we have this column here, original sample name. Again, that's another column that you don't really have to worry about. So now that we have our metadata pulled in, and I left these questions in here just to kind of get you thinking about your data set, right? Because it's going to be a little different. I modified these questions because before they were all very specific to GLDS 104, but now you're going to be doing any data set, right? And so I modified these just to kind of get you thinking about the information that you're looking at. So now we have our metadata. The next thing that we need is the raw counts files. And so we need to download those genes.result files that are generated from RSEM. And so if you remember, those files are also located under files in our repository, right? So in the raw counts data, we see these genes.results files. Those are the ones that we want to download. And so what we do is we're going to use, again, that same Python script. And this time, again, we're going to give it, the same OSD number, so the same identifier for your data set. Except instead of an is.zip file now we want all files that end in genes.results. So instead of is.zip here, we want genes.results files now. And then where we want to put this is, remember, we set up a subdirectory for where to put all of our counts data from RSEM. And so we want to direct all of these downloaded genes.results files and put them into our counts directory that we created above. And so we'll go ahead and run this. And again, you'll notice that it is letting you know that it's pulling each genes.results files for each sample. And it gives you, it uses, again, that curl command to pull that file. It tells you where it's getting it from the internet. It tells you information about like the download speed and how fast things go. And so this, again, runs pretty quickly. If you have a ton of samples in your study, it'll take a little longer. So all of these code blocks, depending on how many samples are in the data set you select, may take more or less time. And so again, just double check that it finishes here and you see a number populated. Make sure you don't get any error messages. It should look similar to this. And then the next thing is to take a look at your counts directory and make sure you see all of those files. So just that, again, that ls command, this time looking at the counts directory. So a couple things that we want to know here, right? So all of these files, again, should look just like the files that we're seeing here. And when we publish files in our repository, we have an automated system to add in the GLDS number, the assay type, this is RNA-seq, and then an underscore. So this is the prefix that's going to be added to all omics data files for a specific GLDS. This information is great, so that way if you're like downloading files from a bunch of different data sets, you'll be able to keep track of which files, which files they came from, or which data sets they came from. But of course, for our purposes, when we're just looking at one data set, we really only want what the sample name is, right? So ideally, we would want these files to start here. So we're going to do a command where we basically remove this prefix from all these files. But before we do that, an important thing to do is, just as a double check to make sure that you downloaded every single file from every single sample in that study, you want to list all of the files in single file. So if you remember from last semester, it's the ls and now the -1 option to list all files in single file. We're going to list all the files in our counts directory that end in genes.results, all the files that we just did, and then we're going to pipe that into a function called wc-l. And this function wc will do a word count, and then the -l says, instead of doing a word count, count each line. So we're going to list all files, one per line, and then we're going to count how many lines, and that'll tell us how many files. And if you remember from last semester, this data set has 6 ground control samples and 6 flight samples, so we should see a total of 12 files here. So this is a good check, and you can go on the repository, see how many samples you have for the RNA-seq assay, and just make sure that you're downloading all the files for all the samples. And so now this is where we're going to get rid of that prefix. So this find function is really cool because you can find a file that has a specific string or a specific set of text in the file name. So for us, we're going to find within our counts directory a name that contains genes.results, that ends in genes.results, right? So we're looking for all the genes.results files. And then sed is a really awesome, very powerful command in Unix. It can do a variety of different things. And basically, I Google, how do I do this with sed? And then Google tells me, and then I try it out and see if it did what I wanted it to do. So that's how I ended up figuring out what this code is. And so what this is doing is it's taking, using the sed function to basically look at this list of files that end in genes.results. And anytime it sees whatever the GLDS number is for that, right, held in the variable GLDS, to take that, and if you remember, I said that each of the file, when we upload it to our repository, it automatically adds a GLDS number, and then an underscore, and then the assay type, which for us is RNA-seq, and then another underscore before the sample name. And so we're just telling sed, hey, anytime you find this prefix, I want you to replace it with nothing. And then this xargs is another really useful function in Unix. It's basically just saying go through the entire directory recursively and change all of them. And so we would run this. And that should be replacing, if you remember up here, it should be replacing this with nothing. So that way it basically is removing this from all these file names. And now you want to make sure that it actually did that. So then in this next code block here, we're going to list what's in our counts directory again. And this time we should see those same files, but with that prefix removed. Now, it's true that all of the files for RNA-seq should have this prefix. However, I can't promise you that sometimes it's not a capital S in the seq or a capital R in the RNA. And so I'm pointing this out because when you list this, if you see, for instance, that the prefix is underscore RNA underscore capital S here for the seq, then what you would have to do in this code block is change this to a capital S. So just make sure that this RNA-seq, the caps, is consistent with what you're seeing in your initial list. And so once you are sure that that prefix has been removed, now we're ready to go. And most of the rest of this should be very familiar to last semester. So we'll probably be able to get through the rest of this pretty quick. Because again, it's going to be very, very, very similar. So now we have our metadata that we all have in that run sheet. So we know all the information about each sample. We know what the factors are for each sample. We have all of our raw counts data from RSEM. So now, pay attention, the kernel changes. And so starting at step 2, we're going to change to an R kernel. The R kernel that we use for this is down here. So when running steps 2 through 5 of this notebook, you want the R kernel that looks like this. So cond environment GL4U-R-2023. So it's an updated version of R from the one that you did last semester. So this is where you want to click on your kernel in the top right. You want to do the drop-down menu here, and then you want to go down to your R environment GL4U-R-2023. So make sure you're selecting that kernel specifically. And then once you have that, click select. And then you want to give it a sec and make sure that up here, you're now seeing that it says R cond environment : GL4U-R-2023. You guys all both seeing that?
Yep.
Awesome. Okay, so now again, very similar to last time. In R, we need to load all of the libraries or all the functions that we're going to be running. So this is similar to what you saw last time. We're going to use tax import to pull in the information. Whereas before you were pulling in a raw counts table, now you're going to be pulling in those genes.results files. So tax import can handle a variety of different file types, including the genes.results files. We're going to be loading DE-seq2, tidyverse, and stringer. These are all libraries that this duplicate stringer doesn't need to be there. So, and then this is just letting you know that it's installing things. You might see some warnings here. Warnings are fine. Errors are a problem. So warnings are no thing. Make sure that it finishes running. I will get rid of this one. So this should all be complete. It'll look like this. It's basically letting you know you might see some conflicts and stuff. Nothing to worry about. We already did load stringer, so you don't need to load it before. It's in this list. I'm sorry. I'll get rid of that in the one. See, we loaded it here. So there's no need to load it twice. It should already be loaded. So you guys can actually get rid of that. I'll get rid of it in the main one too. And then the next set of libraries we're going to load, these are all the libraries that we need to do data visualization. So again, these should look very familiar from last semester. We do have some newer ones. These are all for the gene set enrichment analysis that I added on to this one. So go ahead and press play. And again, you're going to see a series of messages as it's loading in each one of these packages. And then just make sure it completes. And then once it's complete, if you remember, before we were in the bash kernel, and so any variables that we had in bash were cleared away when we started our R kernel. So what we need to do now is we need to set up variables in R. And so the only thing that you'll have to change in this code block is just the OSD and the GLDS number. So that's it. So whatever data set you're doing, just making sure that in this code block, you're changing the OSD and GLDS number. And then we're doing a very similar thing to what we did in the bash kernel. We're setting up variables for OSD, for GLDS, and then for the paths to all of the directories that we set up. So you can go ahead and run this. And that'll run real quick. And then I have you check your run sheet path. So the one path that we added here was a path specifically to your run sheet file. And so go ahead and press play to this one. And you want to make sure that your run sheet path, or that your run sheet file here, the name of it, matches the run sheet that you made in the metadata directory in step 1B. So this will let you jump back up to step 1B. And then if you just scroll down to where you listed your metadata directory, you just want to make sure that these names match. So again, if they are different, so this is where sometimes, like, one thing I ran into is, for some reason, sometimes this S is capital, sometimes it's lowercase. So that's something I'd look out for. If there's an updated to the version of the DP tools, maybe this version will change. So just make sure you're keeping track of the caps, the versions here. And so when you go down to your R, and after you import that and you list that file, you want to make sure that this matches exactly what's in your metadata directory, or else things will break. If it's different, so if this was a lowercase s in here, when you're defining your path, you see how it's ending in that. So this is where you would change it. So like if, for instance, you did ls in your metadata directory and this was RNA lowercase s for seq, then you would go in this code block and just change that to a lowercase. If you saw that it's v2 instead of v1, you would come here and change this from v1 to v2. So just make sure that those match. Once you do that, so here, what we are doing is we are creating a data frame that contains each sample and its factor. So remember, for DE-seq2, if we looked at the run sheet, we had like a variety of different information there, including the factors. And so here we want to kind of look at that run sheet, but only pull out the columns that we need specifically for differential expression analysis. We need the sample names. We need to know all the factors that are there. So that's what this is doing. It's pulling in that information. So the first thing it does, and you can kind of look at the annotation for this, is it creates a function and then it runs a function on your run sheet for your specific data set. So details here aren't really that important. But yes, you can go ahead and, I don't know, I just skipped up to the top. Sorry about that. After loading libraries. We're here. So yeah, you can go ahead and run that. And so what that's going to do is it's going to ultimately, this last part here, is it loads that information that it pulled out of the run sheet into a variable called study. And so this should start to look a little bit familiar. So if you remember, in last semester, we created a variable called study that had the samples listed in one column and the groups listed in the other column. So we're getting to there, not quite there yet. But this now, if you run this and take a look at the study variable that you just made in this code block, you'll see the sample names, and then you'll see this is factor 1, right? So if you had a data set that had multiple factors, you would see a column for each factor here. This just has one factor, So you're able to see just the one factor column. So now the next thing that we want to do here is now we want to create groups. So again, what we're going to do is going to look very similar to this for this one because there's only one factor. In any other data set that has multiple factors, what we're going to do is string together each of the factors using an and symbol or an ampersand. So that's what this code block is doing, is it's creating groups. Because remember, you want your samples to be grouped based on having all of the same factors, right? So if you had radiation and simulated microgravity, the samples that had both of those things you would want in one group. So this just strings them together with an and. Again, it's going to look the same as what it looks like before here because we only have the one factor. So we go ahead and run that. Should run pretty quick. Again, all of these will take longer. The more factors you have, the more samples you have. And then what we do here is we stored all of this in a variable called group. And so now we could just take a look at the group. And the other thing we did here is we associated the order of the samples with which group it's in. So what you see here is the first sample is a ground control sample, and it should be labeled as ground control. Second sample listed was a ground control sample labeled as ground control. And just if you remember right here, first sample listed ground control. So it's just telling, it's basically ordering it. Hey, the one in position 1 is ground control. The sample in position 2 is ground control, like that. So that's what that's doing here. And now what we're going to do is our differential gene expression script is set up to run differential gene expression in all different groups, right? So again, in here, we just have 2 groups, flight and ground. And we run differential expression doing flight versus ground, and then also in the opposite direction, ground versus flight. If you had, say, 3 groups, right? You had group A, group B, and group C. What this would do is it would set up contrast to do group A versus group B, group A versus group C, group B versus group C, and then all of those in the reverse direction. So that's what this is doing. Again, set up like this, so that way no matter what data set is, no matter how many groups you have, it'll create all of those comparisons for you. So you can go ahead and press play for that. And this holds it in a variable called contrast. And so we could view our contrast table. And if you remember, this should look pretty similar to the contrast table that you made last semester. Of course, these names now are pulled from the metadata. So ground control, ground space control, space flight, that's because that's what that factor was named. So that's where that's coming from. So depending on how your factors are named in the metadata on the repository, that is what's going to show up in your comparisons. And then just so you, this is set up to do ground control versus space flight, and then in the opposite direction, space flight versus ground control. And so this is just saying what group is going to be in the first spot in this comparison, what group is going to be in the second spot. So I always like to check this to make sure, okay, this is ground control versus space flight. So ground control should be first, space flight should be second. This is space flight versus ground control. Usually space flight first, ground control second. And then again, I ask you questions just to have you think about the information that you're generating. And so the next step here, again, should look pretty similar. So here we are pulling in the raw data, except instead of pulling it in from a counts matrix like you did last semester, we're pulling in these genes.results files. So we're going to go ahead first and list all of the genes.results files. And then this, we're making sure that the genes.results files that we list are in the same order that the metadata is. And that way it makes sure that it matches up the right counts information with the right samples. And so we'll go ahead and run that. And that's stored in a variable called files. So now if we take a look at files, what we should see is our genes.results files. And this should be set up so in the samples should be in the same order that we saw the samples in this table. And so that's all of our files. So, okay, we pulled all of those into R. You're basically reading them into the R environment. And now that you've read them into the R environment, we're going to put them in a tax import object. So this is what we did with the raw counts table. So remember last semester, you pulled in the raw counts tables into R, and then you put it into a tax import object. We're going to do the same thing here, except this time the type is RSEM. I think last semester it was called matrix because you were pulling in information from a matrix. This is so tax import knows that these files are RSEM because it knows what to do with that. And then we have to add lengths to genes with zeros. It's just because it'll throw an error if there's a gene length of less than 0. So go ahead and run that. And it'll tell you that it's pulling in each one of your samples. So the numbers here should correspond to the total number of samples in the data set you're doing. Now that we have our samples and our counts data, what we want to do is create a sample table that contains the information of our samples and all of our groups. So you run that, you store it in a variable called sample table, run your sample table, and this should look very similar to what you saw from last semester, making your final table where it lists all of your samples and it lists all of the groups. So again, we only have one factor here, so this looks very similar to the table above where we pulled in factors. If you had multiple factors, the condition, which is the group, it would be ground control and whatever the next factor is, spaceflight and whatever the second factor is. So basically, these would be stringed together with ands if there's multiple factors. Just prompting you again for some more questions. Now, that's pretty much all the parts that had like differences until we get to the end from last semester. Now we have all of our metadata, we have our sample table in here, we have our counts data imported into tax import. All this is going to look super familiar. So we'll go through this quickly. The first thing we're going to do is create that DESeq dataset object from tax import using that object that we created that contains all of that RSEM raw counts data, the sample table that we just made above right here. And then it's telling us to create the dataset object using the condition column, which is our group column. So that's what this is doing. So we can go ahead and run that. And then similar to what you did last semester, let's take a look at what this DESeq dataset object looks like. And if you remember, the number of genes will tell you in the length of this object here. So right now there's 55,000 genes. Some of those genes are duplicates, so there's not 55,000 genes in a mouse, as far as I know. Less than that. And so again, we do some filtering steps here, just like what you're familiar with from last semester. So we move really low expressing genes. Any gene that has a sum of less than 10 across all samples, we're going to remove it. And then we take a look at our new DDS object that we just filtered. And now it went from 55,000 something genes to 21,700. So just having you think about how many genes were removed because they were low expressed in here. And then we can look at the dimensions of this object. And this should be the total number of genes after filtering that we just saw above, and then our total number of samples. And now we're ready to normalize the data, right? So at this point, we still have raw counts data. We imported into a DDS object so that DESeq2 knows what to do with it. And now we're going to normalize it. But before we normalize it, just like we did last semester, let's take a look at how the samples separate based just on the raw data. So before we do any normalization. So we're going to go ahead and create a raw counts data frame, which is basically stringing together the raw counts from RSEM into a variable called raw counts. And then we're going to import those raw counts perform the principal component analysis. So we take the log2 of the raw counts, and then we added a +1 codes just in case any raw counts is 0. 0 would give you an error when you take the log of it. So we add a 1 to all raw counts. And then transform it into log2, and then use this pre-comp function for principal components, and we store that object in the PCA raw. So again, should look familiar from last semester, and then take a look at the summary of that information, and this shows you all the principal components, again, grouping the samples based on similarity of the raw gene expression in this case. And so then we're going to create a raw plot, raw PCA plot here. So this is using the raw data. I have this set up same as last semester. So first we create a plot where all the samples are labeled. This helps if like there's a sample that's really weird, you can kind of go back into the metadata and see, oh, did it have a really high RIN, or did it have a really low number of reads, or something that would make sense of why it's an outlier. And then we can save that file with the ggSave function. And this is going to save it into the PCA directory, that the variable that we made for the PCA directory in our structure. We'll take a look at all the output files after. And then we create another plot, same exact thing as last time, except we just have those sample labels removed. So nice and clean here. again, using the GG save function. If you are going to be creating these files for like a poster or something like that, I just want to point out that, remember, you could play around with the width and the height. So when you go and find this file in your directory structure, if it's like things are cut off or something like that, you could make it wider or taller by changing these parameters here. And if you have any questions, like when you get to that point and you're really ready to make these pretty for like a poster or something, and you forget that, just let me know and we can have a quick call. And then this is just that PCA table that we took a look at up here. We're just printing out that PCA table to a file in this last code block in this section.
Amanda,
Yeah.
how do we export the tables? I mean, figures,
Mhm.
or we just use print screen?
So you actually did it with this command here. So this GG save function, that actually saves the most recent plot that you made. So with this auto plot, that's which generating the plot that you see here. And then this line here is saving that plot in the file path that has the PCA directory. And then we're labeling it with the GLDS number and then with underscore PCA raw. And this one didn't have sample labels, so no labels. So then that plot you can find if you go in your directory structure here under 04 in PCA. You should see this is where the plots were labeled. They're PNG files, right? And you can right click and download that to your local computer. And then when you
Right.
download it, when you download it, you open it up, take a look at it. And that's where if things don't look right, you could basically play with the width and the height of the file that you're generating, and then take a look at the new file that you saved. And, and then you could, keep playing with that until you get something that looks good. Uh, and if you want to make sure that you kept the one that you made before, just make sure you change the name, add an underscore here after the no labels to _1, _2. So that way you're not overwriting the file that you make each time. And
Thank
again,
you.
if you have any trouble with that, when you get to it, just let me know, and we could hop on a quick call and I could go over that.
Thanks.
Yeah, so that was all the raw data, but raw data doesn't mean much because we haven't normalized for differences in read depth. We haven't normalized for differences in gene length. We haven't normalized for a potential RNA contaminant or something like that. So now we want to run through the DE-seq2 normalization steps. And so, that's a series of 3 steps from last semester. So we estimate our size factors, and we're storing this in a variable called DDS1 now, just to distinguish it from the original DDS object. And then we're going to do the estimate dispersions. And then just to take a look at the dispersions plot and how they changed after this normalization. This again should look familiar from last semester, where we have the original gene set in black, and then it generates this fit map here. And then after it does the fitting based on the data, you'll see the final in blue. So basically just coming tighter around that line. And then the last thing that we do is the hypothesis testing using the wall test method. So we run that. And now that finishes the DEC2 analysis, and we should now have normalized counts. And so we're going to pull out the normalized counts from this DDS1 object, and we're going to store the normalized counts as a data frame in a variable called norm counts. Again, similar to what you did last semester. So you go ahead and do that. And now that we have our normalized counts, the other thing that we did here, and I don't know if you taught this part or not, Jen, but we do a likelihood ratio test too. So this is doing essentially the norm wall test is doing essentially like similar to a t-test, comparing each group. And then what also you could do is a likelihood ratio test, which is a slightly different type of analysis that you can do across all samples here. So we're also going to generate the likelihood ratio test, which is going to give you a LRT value, likelihood ratio value.
Sounds familiar, but I'm not sure if we went over. If it's in the Jupyter Notebook, we should have gone through.
Yeah, it's not in that one. It was something that we did during the boot camp. I think someone asked about it and wanted to know about the likelihood ratio test. So I added in here.
See.
You could do it or not.
Okay.
It'll just add an extra column in your final DGE table. And then if you wanted to look at that statistic, someone asked about that statistic, it would be there. So now what we're going to do is create another PCA plot, but this time using the normalized counts. So remember, we normalized the counts, we saved it in a variable, the normalized counts in a variable called norm counts. So then we're going to go through and take the log2 of those values, similar to what we did with the raw counts. And then we're going to calculate the principal components and store that in a variable called PCA norm. So we can go ahead and run that. And then we're going to plot the normalized PCA. And so again here, what you tend to see, if you noticed from last time, that what we see is a larger value in PC1, which is where the samples separate based on the biological groups. And we actually see a little bit tighter in PC2, which is any differences between samples. And you'll probably notice this using the different data sets that you do for space flight. It tends to be more variable than keeping it on the ground. Again, just individual differences of the organism that you're working with. And then just asking you to think about the data. And then this is just giving you some extra code blocks. So if you wanted to play around with some of these variables, like make these dots bigger, you could change this size. Alpha is transparency. So that's a number from 0 to 1. So let's say in your data set, you had like a group of circles. It only looked like you had like 2 circles there, but really it should be like 10 samples. It might be because they're all overlapping on top of each other, super tight. And that's where I would change this alpha to like 0.5, and it'll be more transparent. So you could try to see if there's more than just what's there from doing it with the bold color. And then after that, now we're all into the differential expression analysis. So of course we ran that with DE-seq2, and this is basically just pulling in the information from running DE-seq2 and putting it together into a table. And so we start that table with our normalized counts. And so basically this is just saying, hey, let's set up another variable and let's have that variable contain the normalized counts. And then we could take a look at it. So here's just looking at the head of the table. And so again, what we should see is our ensemble IDs here on the left. And then this is the normalized expression for each sample for each one of these genes. So you should see a column for each sample. We have 12 here because we have 12 samples. If you have more, you'd have more columns here. And then here I'm just having you, because we're going to go through the wall test now. So we'll take a look at the contrast again. And then what this does is it's going to iterate through each contrast, because when you ran DE-seq2, you specified the condition as the column, or you specified this is the condition. And so remember, this, we only have 2 groups, right? 2 unique conditions. In data sets that you run that have multiple factors, you might have 3 groups, you might have 4 groups, you might have 5 groups, it depends. And so what DE-seq does is it'll take each unique condition and it'll run those pairwise comparisons for them. So what we're doing here in this code block is we're actually going to pull out the information from each comparison from every contrast that was analyzed when we ran DE-seq2. And so that's what all this code chunk does. And this will, of course, take longer the more information that you have. And it's also pulling out specific information about each contrast. So it's pulling out the log fold change when you compare one group to another. It's pulling out this Wald statistic, which is similar to like a z-score. It helps you rank genes based on how different they are in your 2 groups. And we're actually going to use that in the gene set enrichment plot, which is pretty cool. And then we also pull out the p-value, which again doesn't take into account false discovery rate, which is very important when you have, what do we have, like 20,000, over 20,000 genes. That's over 20,000 comparisons. We really want to take that into account when we're running our statistical analysis. So that's why I say never, ever, ever go by the p-value to determine significance with RNA-seq data. Always go by adjusted p-value, which adjusts for multiple comparisons, since we have so many genes. But we do pull out both those columns in the table. And then we're going to perform some calculations here. So we're going to pull out some of the calculations here. So first is all mean. So this is just the average for each gene across all samples and the standard deviation for each gene across all samples. And then this is just giving you a challenge. So you want to take a look at what the DGE table looks like now. You could just type in head here and then the DGE output table and run that. And you can kind of see the columns that we added here at the end. So we pulled out the log2 fold change for each comparison, right? So we have ground control versus space flight, the log2 fold change, the wall statistic, the p-value, the adjusted p-value, and then we did space flight versus ground control. Again, log2 fold change, statistic, p-value, adjusted p-value, and then we also pulled out the average across all samples, and then the standard deviation across all samples for each gene. And so we have one comparison here. So we have two sets of data, right? So we do the comparison in both directions. This gets really big the more groups you have, because it'll do pairwise comparisons in each direction. So this could get crazy big. Just a fair warning about that. It can sometimes be overwhelming. So the next thing we're going to add is that likelihood ratio test variable that we calculated, which I know you guys didn't do last time. So it just adds another column to the table there. And then this, you can go ahead and run it because it takes a while, and then we'll talk about it. So what this is doing is it is calculating the average expression for each gene in each group. So basically for every single gene, it'll look at all the samples that are in the spaceflight group in our data set and take the average, all the samples that are in the ground control group and take the average, and then it'll do the same thing, and it'll take the calculated standard deviation as well. So how long this, and then it's going to add the columns to the DGE table. That's the cbind command. And how long this takes will, of course, depend on the number of samples. And you'll notice here, I think it takes like a minute or a minute and a half with just 12 samples in two groups. So just bear in mind that you might be able to start this, go run, grab a snack, come back, depending on how big your data sets are that you pick. And then again, I prompt you for questions just to take a look at your data and think about it. And this will also help, like if you submit your results for an abstract at a conference and you have questions from someone who comes and asks you to explain things, hopefully if you thought about this, looking at these questions, it'll help you answer those. It does look like, I know we have only about 6 minutes left. I'm probably going to need 10 or 15 more minutes just to be able to go through the GSEA plot. I don't have anything after this. Are you guys okay to stay on an extra 15 minutes?
Yeah, that's
15
fine.
minutes, okay, but I have a class at 1.
Okay.
So
Yeah,
15
we'll definitely,
minutes.
yeah, we'll definitely be done before 1.
Sorry. Yeah.
No, that's perfectly fine. I always underestimate this time. It's because I'm a talker.
But that's
Obviously.
great. We love it. Yeah.
Are both of your guys done too? Are you still waiting?
Yeah, it's finished.
Excellent. And so now that this is finished, it was added to the table. Basically, this again is prompting you if you want to go ahead and take a look at your DGE output table now, you should see those group averages added. So you could just run head command here again and take a look. And these are all just, every time we add stuff, we just kind of add it to the end of the table. So you have, of course, all of your comparisons with your stats, your all averages, the LRT, this is a likelihood ratio p-value that's new for this one. And then we have our group means. So for ground control and for spaceflight, the average expression for each sample, and then the standard deviation for each sample for each gene, or for each group for each gene. And so here's another different part. So this is what I was talking about, where instead of pulling the information from bioconductor and then pulling it from Panther and then pulling it from StringDB, we're just going to use that CSV annotations file that we looked at from our repository. So this set of code block is basically pulling in this information to add these annotation columns to our table for whatever your organism is. So if you want to learn more about that, so this is just a link to the Open Science Data Repository. This is a link to that CSV annotations table that we went over here. So it links right out to this table. And then this goes to the top level directory for that GeneLab reference annotations. And so the cool thing about this is if you remember in the run sheet file that we made at the beginning, do you remember one of the columns was organism? And then in this table, we have a column called species. So that's what it's mapping to. So it's actually looking for the organism name in the run sheet that we have is the species name. So I probably should make these consistent, but so it's looking for the species from your run sheet. So you don't even have to put in this information in because it's in the run sheet that you already generated. So it's actually looking for that column and it's matching for ours. We have Mus musculus. So what this code block is doing here is it's basically taking a look at your run sheet, mapping it up to this table here, matching up the species. So it'll match up this column here, and then it'll know that, okay, I know that your species is Mus musculus. That means that the annotation table that I have to pull is going to be this annotation table here, which is linked to the mouse annotation table. And so it'll do that for any organism. So let's say you're doing like a Arabidopsis, which is a type of plant. It would look for Arabidopsis here, and then it would pull in this table, which is going to look similar to the one that I apparently just X'd out of, but it's going to have annotations for Arabidopsis instead of the mouse. So yeah, we can go ahead and run this. So that's really all that it's doing, is it's just pulling those annotations. And then it is pulling it into a table called annote. So if you remember, that's the annotation table that we made last semester. And so this is just a challenge for you to take a look at the head of that annote table that we just pulled in. And it should look just like what we had before. So the ensemble ID, the symbol, the gene name, we have these RefSeq IDs. We also have modified this. So if you remember before, I think the RefSeq ID was only one. So we would only take like the first RefSeq ID, but there's a lot of RefSeq IDs associated with these genes. And so these RefSeq IDs could be a gene reference, it could be a protein reference. And so we decided, let's have all of them in here. That way you don't have to go looking for them for a specific gene. So they're all separated with a pipe. And then we have the Entres ID, which is just one number, and then the string ID, which you remember from last semester. And then this is the gene ontology. So if you remember, there was that long list of gene ontologies. And because gene ontologies are basically, right, like a group of a particular type of biological process or molecular pathway. And so there could be a gene that is associated with multiple different biological processes, right? So our bodies are very efficient, and we can use the same gene depending on where it's located in our body to do a completely different function. And so that's why you have multiple gene ontologies associated with a lot of the genes. And we're going to use those actually to do gene set enrichment analysis. And so now what we're going to do basically is this is where we combine the tables. So we have this DGE output table that just has the ensemble identifiers. And now we have this annotation table that has ensemble identifiers mapped to like the gene symbol and the gene name and the gene ontologies. And so this is what this is doing here, is it's basically merging that output DGE table that we've been building with the annotations table that we just built. And it's saying to merge them based on row name. So it's merging them based on the ensemble IDs. One thing I do want to point out here, if you are working with a plant organism, the ensemble ID, it actually does not have an ensemble ID, even though we're pulling from the ensemble database, which is a little weird. The IDs are called TAIR. And so I have this note, it's noted several times. So just if you're working with a plant data set, anytime you see ensemble in the code block, you'll have to replace it with TAIR. And it's noted all throughout here, but just wanted to point that out. So we'll go ahead and merge those tables. And then we're going to take a look just at the first row of the table. And so just at the first row, now we have the ensemble ID, and now we have all these annotations added. So the symbol, the gene name, RefSeq, Entres, string ID, our gene ontologies, known as GhostLim IDs, and then all of the raw or normalized count data for each sample, and then, of course, all of our statistics that we had in the DGE table. And if you forget what each one of these columns are, I left in here the definition of what each of these columns are. So if you're ever confused about what information is in here, you can take a look at here, and you should get a good idea of what it is. And so now what we're going to do here is we're just going to output these tables. So what this is doing is it's outputting that sample table.csv file. So that's the table that has your samples and which group each sample belongs to. This contrast.csv file, so that's all the pairwise comparisons of all the groups. And then your differential expression CSV file, which is the table we just looked at, that contains your gene annotations, your normalized counts, and then all the DGE stuff at the end. And so now that we've put all this information together, have a nice table, we can use that information to start visualizing our data, which is the most fun part of RNA-seq. And so here, one of the first things that we're going to do is we're going to log transform our normalized counts. And now remember, when you do your data sets, your group names might not just be spaceflight and ground control. There might be a variety of different groups in there. All of them are going to be generated in a pairwise manner. Whereas last semester, and in this example, we only have the spaceflight and the ground control. And so I just used FLT and GC in all the visualizations from last time. This time I want to make it more flexible. And so what you want to do here is you want to pick out which groups you want to compare. So remember, our script is set up to do pairwise comparisons of all groups. Sometimes all pairwise comparisons don't make sense, right? So let's say you want to know how radiation changes with radiation exposure and without radiation exposure, but you also have this other variable of simulated microgravity. Well, looking at radiation compared with simulated microgravity is not going to tell you the difference between with radiation and without radiation, right? So you want to pick which comparisons is going to interest you. And this is something that Dr. Lin can help you with, is to think about what biological question are you asking of the data? What do you want to know the differences for? And so depending on what groups you have, you could decide, and then group one is going to be what you're going to compare it to. So all of this is set up to do group one versus group two. So that means in your output table, anything that's enriched or more highly expressed will be more highly expressed in group one. Anything that has a negative value or more lowly expressed will be suppressed in group one or more highly expressed in group two. So this is where you really want to think about the biology of your study and the question that you're asking. And you select which group you want to be group one and which group you want to be group two. Now in your data set, if you have multiple groups and you want to look at multiple comparisons, you'll run all the next steps doing the first comparison you want to see, and then you'll go back and change G1 and G2 and rerun all of these for the next comparison that you want to see. And so you can run this as many times as you want for all the different comparisons that you have, for just two of the comparisons that you have. It depends on the question that you're asking of your data. The thing that's important here is you want to make sure that how you name group one and group two is exactly how group one and group two is listed in your metadata. If you forgot, just add a code block here and just take a look at your contrast table again. So if I just add a code block, play my contrast table, this is what one of my group names look like. This is what another one of my group names look like. Make sure you're going based off of here, because how R plays with things, you see it doesn't like spaces, so it'll add a period. But what this is looking at is your DGE table, which is going based off of the column headers here, right? And so for us, for spaceflight versus ground control, I usually like to look at spaceflight on top, ground control on the bottom, so that way genes that are upregulated will have that are upregulated in space will have a positive value. So here I wanted my G1 to be spaceflight, so I literally just copied this and pasted it in here, and I wanted my group 2 to be ground control. I copied it here. So this contrast table, again, could have like 20 different contrasts. So you'll just scroll through your contrasts, find the contrast that you want, and then copy the group 1 into here, copy the group 2 into here. So that's as far as you have to, and then everything else stays the same. So now the first thing you do is, of course, run this to set your variables of G1 and G2 to whatever groups you want to compare. And then what you're going to do here is similar to what you did last semester. We're going to filter this now to only pull out differentially expressed genes. The cutoff values that we're using here is a log2 fold change greater than 1 or a log2 fold change less than -1. And so that would find genes that are upregulated and find genes that are downregulated. And the other filtering that we do, which is up here, is we filter the adjusted p-value to only grab genes that have a significance of less than 0.05. And again, you could play with these. You want to be more stringent, you could increase this number. You want to be less stringent, you could decrease this number. I think this is a really good starting place. And then depending on what your data looks like, you might want to play with these values and see what your data looks like. So all this stuff is going to be kind of up to you based on what your interpretation is of the science. And so now you could take a look at your dimensions. So if you remember, that initial table had like 21 something thousand genes. And now after we filtered out for just the significant ones based on these values, we see 734 genes. This is the rest of the columns, right? So it's the 12 samples plus all those other column headers that we have here. And so the next thing that we want to do here is set this up to be able to run the PCA plot with just differentially expressed genes. And so here, again, just want to point this out because now we are going to run this on that, but we want to get rid of all those other columns and just have our sample columns because that's all we need for PCA. And so again, this is another place where there's ensemble. So if you're using a plant data set, make sure you change that to TAR. And so you go ahead and run that. And now you should see that same number of differentially expressed genes, but now only 12 columns because it's just your samples, the normalized data for those differentially expressed genes. And now you're going to use that to create a PCA plot of just the differentially expressed genes. So similar to what we did with all of the normalized data, except now it's just the differentially expressed genes. You're again going to do the auto plot to make the plot of this, and then you're going to save it with that function down there to save. And then what you should notice here is that this number should increase a lot, right? Because we're only looking at genes that are differentially expressed in our two groups. So we should see a big difference based on the data that we have here now. And then again, just getting you to think about that, pretty much what I just said about that. Next is a heat map. So again, this should be very similar. So we're going to go ahead and do transform the data between 0 and 1. You remember, if not, it's because, sorry, I'm getting over sickness. If it's because these genes could have a very wide range in expression, they could not be expressed, and they could be expressed at like 100,000 counts. And so we really want to kind of get them all on the same playing field, or else making this plot would be impossible. And so you just go ahead, excuse me, and run that, and you'll get your heat map for your groups of interest. And then here again, it's the same thing, right? We're just going to save it. So now we're just storing it to a variable called heat map. And we're doing that so that way we can use this save PDF function to save your heat map. Again, this is going to be in the heat map subdirectory within your differential expression folder that you made. So you'd go ahead, download that file, take a look at it. If it looks weird, if you want to change the width or the height, you'd play with these width and height parameters. So same goes for all those plots. And then this is just reminding you that if you don't scale that between 0 and 1 or take the log function, you'll crash the kernel. Next is volcano plot. Again, this should look very similar. And here again, too, you could play with your cutoffs. So here I have a cutoff of 1 for log 2 fold change and a cutoff of 0.05 for adjusted p-value. So you'd want to change it in your label, and you'd also want to change it in the actual p-value and cutoff here. So play that. It should look similar to what you saw last semester. All of these plots are labeled. So one thing that I kind of have here in this challenge is I added this additional thing. So let's say you're not really interested in all of these genes being labeled, and it's kind of messy, and you found like a pathway that's really cool, and you only want to highlight specific genes that are in that pathway in the figure that you create. You can actually take your gene symbols and make a list of there. So if you look at the documentation here, it'll bring you to the documentation for the enhanced volcano plot. And so, for instance, you could add this row here into your command here. So basically, you just copy and paste this. Do it here real quick. And then let's say I'm only interested, so copy and paste this line here. So this is selecting specific gene symbols to label. Whereas here, it's going to auto decide which ones to label. So just make sure you start in the same indentation, except let's say you only want to know KRT17 and SLC4A3. So then you would just say, all right, I only want to label KRT, make sure your caps is the same, 17. And you could pick from the gene symbol column in your DGE table, an SLC4A3. So now I run this again. And now instead of all of the genes being labeled, just the ones that I told it to label are labeled. So you can kind of customize this. And then there's more things you could do. Like if you don't like the axes that are automatically generated, You could take a look at that documentation file and it'll tell you what to add in that command to like change your axes and things like that. So this is the new one. That's everything that you've done from last semester. This is gene set enrichment analysis. So what gene set enrichment analysis does is it's basically going to take gene sets, right? So sets of genes that are associated with, depending on what your gene set is, it could be associated with anything. So here I have this set up to do gene set enrichment analysis based on gene ontologies. So if you remember, genes can be associated with a variety of different gene ontologies. Gene ontology is just a way to group genes based on their biological function. So genes that are involved in breathing, right? Your airway pathway could be a gene ontology. And then it'll have a list of genes that are all associated with your airway pathway, right? And so one of the first things that we need to do for the gene set enrichment, because one thing that it takes in is a bioconductor package for annotations for your organism. Again, this information is in this table. So for mouse, this is the bioconductor annotation package, the org EGDB here. And so what this script is doing is it is looking for this database from this table. So that's all this is doing here for your specific organism. And it's saving it as a variable called ann.dbi. And then here, I know we're coming short on time, so you could filter this in any way you want to. So you don't have to have a cutoff here at all. You could have this as 0. Or sometimes it's nice for these to like get rid of, I know we got rid of some low expressing genes, like with the sum less than 10, but sometimes it's nice to get rid of, some more lower expressing genes. And so in the default settings they have for this, it's 50. So basically any gene that isn't expressed at 50 across all samples, get rid of it, because it's kind of low expressing, which, might be an artifact. So you could change this more or less stringent, again, however you want. The definition of which each one of these things are here. The other thing that we want to do is for gene set enrichment analysis, you want to rank your genes. So you can rank your genes on a variety of things. So you can rank your genes on log2 full change, right? So that way you set your gene list to have the highest log2 full change on top and the lowest log2 full change on bottom. You could set it based on p-value. Again, I never recommend using p-value, but I have it here as an option if you want. Or adjusted p-value. So you can rank it to have like low adjusted p-value to high adjusted p-value, right? So really significant to not so significant. What I recommend is the Wald statistic. So this is similar to a z-score. It basically just says for each gene based on all the other genes, how much is it contributing to the difference in the log2 full change value? And so I like to go based on that. And so I put stat here, but you could change how you want to rank your genes by copying and pasting any one of these things. And then the next thing is the key type. This is just so gene set enrichment analysis, the algorithm knows what IDs you're using. So again, this is ensemble for all organisms except plants. If you have a plant, make sure you change that to TAR. So again, we're setting these cutoff values as variables. So that way you could just set it up here and then be able to run your stuff for your group comparison. So I think we got like 2 or 3 more code blocks. The next one here is we're filtering all results based on the contract of interest. So basically we're pulling out the IDs, which are our ensemble IDs from our DGE table. We're pulling out the all mean value. That's to be able to filter it based on your filtering thing that you have here. And then we're pulling out each one of these things that I said you could use to rank. So we're pulling out the log2 full change. We're pulling out that Wald statistic column, the p-value, and the adjusted p-value. And then we're putting that all together in another table that we're going to hold in this variable, DGE res. Go ahead and run that. It'd be pretty quick. And then we're basically going to apply that filter. So this mean cutoff filter variable that you defined up here, we're going to take all those DGEs and we're going to filter based on that variable that you added. And again, you don't have to do this. You could set that to 0 and that's fine. But we're going to filter it to get rid of anything that's 50 or less. And so now you can take a look at how many DGEs you're left with. And again, remember, this starts with the full DGE table. So it started with the 21,000 genes that we had in there. So filtering it with ones that are less than 50, we cut that almost in half. So again, you could play with that. And then if you want to take a look at what the table looks like, so again, your ensemble ID, the all means, so these are the columns that we pulled here from that DGE table. And then we're going to take here, what we're doing is we're ranking this table now. So here, just want to point out, we're going to rank it by the stat column. So again, that is specified up here. So you can rank it by log2 full change. You can rank it by the stat column, rank it by p-value. You could pick what one you want here. I again recommend stat, but if that data just doesn't look right to you, you want to do something else, go ahead and try it. And what's that going to do is it's going to rearrange the order of the genes in this table based on the stat value. And so we're going to go ahead and play that. And then I have saved this as a variable called DGE res ranked, and then taking a look at it. Now you see, whereas before the stat column was kind of all over the place, after you rank this, you should see that it's in descending order based on the stat column. And again, because the stat column is associated with how significant these genes are in your comparison, you should see things like the adjusted p-value kind of also going in increasing order, right? Because a smaller adjusted p-value means it's more significant and it would have a higher stat value. It's also pretty nice because the stat value is calculated such that the lower expressing ones will be a negative value in the opposite direction. So if it's really significant but downregulated in spaceflight, it'll have a high value but in the negative direction. And so now here we're extracting basically this list of genes. So your ranked list of genes and the value of the stat column. So that's what this is doing, is just taking these genes and the respective stat column and putting it in a format that's needed by gene set enrichment analysis. So
This is
here's your
very
gene set.
useful. This is very
Yeah,
useful.
right.
I thought I should, we will export it to Excel and rank over Excel, but this is great.
Yeah, don't even have to do that. Do it right here. And
Yeah.
now the gene set enrichment parameters. Now we're ready to run gene set enrichment. And again, this is just going to produce scores based on your sets of gene ontologies. Gene ontologies have sub-ontologies within them, so how the gene ontologies are categorized, basically. And so they could be categorized by biological properties, molecular function, cellular component, or you could use all three. Most people do biological processes, so that's what I did here, but I set this up so you could change it, do whatever you want. Different gene sets will have different sizes. This is just the number of genes in each biological process, right? So some process might have a lot of genes, some process might not have so many genes. If you only were interested in gene sets that had like at least 10 genes, you would set this to 10. And also some gene sets could have like a ton of genes, right? Like, so maybe a pathway that just involves a ton of genes, and maybe you want to set the maximum size. And then you also have a p-value cutoff and a p-adjusted cutoff. Definitions of these are all here. The p-adjusted cutoff will correct for multiple comparisons. You could pick which method you want to use. There's a really nice paper here that describes the difference of these multiple comparison methods. A lot of statistics, I know, something I wish I did more of in school. Highly recommend taking more stats classes. A common one here is the Benjamin-Hutchberg adjustment. So this one utilizes the false discovery rate when it adjusts for multiple comparisons, which is super useful for RNA-seq data. So I recommend that one. That's the one I have here. Again, you could change any of these. The minimum gene set size I have is 10. It's the default for this tool, max at 500. I put a p-value cutoff is 0.05. It did the adjustment value. Oh, this is biological processes. That's this up here. And then this is the type of adjustment. So again, I did the Benjamin-Hutchberg adjustment. And so we just play that to set those variables, and then you run your gene set enrichment. So go ahead and click on that. This takes like 30 seconds or so to run. The thing I want to point out with this, depending on the size of your data set, if it's a lot, if you're the two groups you're comparing have a ton of samples, this could really use a lot of memory. And so if it uses way more memory than we've allocated based on the server that we selected, it'll crash. It'll crash. It'll tell you your server crashed, you ran out of memory, and you'll have to press OK. If that happens, I have this note actually in here. So in here, it's troubleshooting. I have a troubleshooting SMCE server issues. So if you run into an issue with the SMCE server not working, check out this troubleshooting thing. It'll give you some tips. One of it is if you see that this memory goes so much that it actually crashes. I just have a note here to go ahead and spin up a new server, but this time select the extra large server, which has more resources. So for this, you should be good with the server we selected. But again, if it crashes at this point, Just restart your server, use an extra large. If you do have to restart your server, you have to run all the R commands again, because remember, all of your variables will go away. Now that you've run that, you're going to have a GSEA output table. I won't go through this because I took some time writing out the definitions of what each one of these things mean, and I know we're getting close on time. Basically, we're going to pull out the gene set enrichment analysis as a data frame and store it in a GSE table. We're going to save that table, and we're going to look at it. So each one of these columns here is basically a value for the gene ontology. You'll see the description, right? So this gene ontology has genes involved in protein folding. This one has genes involved in ribosome biogenesis. This one in gland development, etc. If you were to have picked molecular pathways, the description here would be molecular pathways as opposed to biological processes. So that's how you can play around based on how you want to organize your gene ontologies and your groupings. If you want to know what each one of these value means, these are basically assessing the value of how important these gene ontologies are in the differences that you're seeing between your groups. So if you want to know more about them, I wrote out definitions here for each one of them. If you're confused after reading this definition, let me know. I'm happy to explain them further. So now that we have this information, we want to plot it. So this first plot that we're doing is called the dot plot, and this is really cool. So what we have here is the biological process. And then on this side, it's biological processes that are activated. And then we have this side here that's biological processes that are suppressed in your comparison. So for us, it's flight versus ground control. These biological processes are activated. These biological processes are suppressed. If you want to know how many genes are in the gene set for each one of these, it goes based on the size of the dot. So the smaller the dot, the fewer the genes. The larger the dot, the larger the genes involved in your data set. This also shows you, based on a color coding, the adjusted p-value. So what we see here is things that are more red are more significant, things that are more blue, less significant. And there's cutoff values that you implemented above with your p-value cutoffs. And so this, I also have this ggSave function. So you'd want to take a look at this in your GSEA subdirectory. And again, if you're not happy with the size, play around with it. The other thing I wanted to point out is we had, it's in this thing here. So I have a note here, adjust the show category parameter for more or less gene ontologies. So here I'm showing 10. So it'll show the 10 most upregulated gene ontologies and the 10 most downregulated gene ontologies. If you want fewer, reduce this number. If you want more, increase this number. So there's a note here to tell you could play with that. The next thing and last thing that we're going to make is a map of our gene set enrichment results. So you can go ahead and play that. This is really cool. So it'll look at all of your gene ontologies and it'll see how they're related to one another and form a network. So gene ontologies that are more, or biological processes, excuse me, because we selected biological processes that are more related will be close together. And if they have overlapping genes in their gene sets, you'll see them connected in your network. And if there are significant, but they're not associated with other genes in the gene sets that are overlapping, then you'll kind of see them just kind of floating here. So here it looks like we do have a little bit of a network with the biological processes that are significantly upregulated in the group for us space flight versus ground control. Again, all these are set to save your output to that output table. And so you again can play with your set here. So this is for space flight versus ground control. Again, if you had another group, another comparison you were interested in, you would just go back to the top of the visualization section, so section 5 here, and you just change your G1 and G2 to whatever one you want next. Go ahead and run this. All of the output files have the group names saved in them. So once you change the group name, it'll change the names of your files, so you won't be overwriting anything from any other group comparisons. And then just to show you quickly what this looks like in the structure, so go to DESeq2. You would go to these are all your tables that we made, our differential expression tables, and then you go to plots, and then it's all separated out into here. So if we look at the GSEA plots we just made, you'll see the name of it. It's the GLDS number, your group comparison, and then it even tells you here that this is the ranked dot plot, the network map, and then that's the CSV file of the GSE output table. And then again, for these, right click, download them locally. Take a look at them, play with the width and height as you will. And then if you want to export this table or this completed notebook, you would just go to File, Save Export Notebook as HTML, and then you would select a location on your computer of where you wanted to save it. Click Save. You don't have to memorize that. That's in this notebook here. So there's export the completed JN as an HTML, tells you how to do that. And then it tells you make sure always that when you're done, you log out of the SMCE server. So instructions do that. File. You can go to Hub Control Panel and stop your server, or you can go to, it's usually good to stop your server. But you can also go to log out. Log out is supposed to automatically stop your server, but I've noticed that sometimes it doesn't. So it's good to go to Hub Control Panel and actually stop your server before logging out. But if you forget and just log out, that's okay.